So welcome everyone to the post-acute sequelae of COVID-19 or PASC seminar series. This started, on October 4th was our first seminar by Michelle Gong, and that was really good. And we had last Friday we had Dr. Frank Molly Jarvis, COVID-19 and diabetes, and today we have Dr. Mohamed Abdel-Mohsen. This series has been, let me go back just to talk about the logistics. We are recording this for transcription purposes, and transcripts from the seminars will be made available for review in the future, and the attendees are going to be muted and will be unmuted for questions and answered, but you can also put questions on the chat, and then we can convey those for discussion. And these talks will also be available for viewing for everyone who's registered. It's been really a great pleasure of mine to work, with the active group, Dr. Joe Manetsky, Courtney Copeland, Trey LaRose, and organizing this together with Dr. Josh Fessel, who's medical program officer at NIH and Heart, Lung, and Blood. Has been a great pleasure, and I think this is a really informative series, and we really enjoyed this so far. So at this point, I'm going to introduce our speaker today, Dr. Mohamed Abdel-Mohsen. He received his PhD in virology in 2014 at University of California, San Francisco, in a joint program with Ain Shams University in Egypt. He postdoced with the Blood Systems Research Institute at UCSF, where he maintains an affiliated investigatorship. Currently, he is assistant professor at the Wistar Institute and University of Pennsylvania, and he is co-director of the Virus and Reservoirs Core at the University of Pennsylvania Center for AIDS Research, CFAR. Dr. Abdel-Mohsen has published extensively on glycomics, glycomic modulation of proteins, antibodies, the microbiome, and its role in interaction with the immune system, HIV, and other systems. He's a young investigator, done extremely well since 2017, when he began his appointment as assistant professor at Wistar and UPenn. He's published over 40 articles of his 65 articles total. He has, I think, 4 R01s. He's also has a foundation funding. He's got editorial responsibilities. He's guest editor Frontiers in Immunology, guest editor MBio, and he's also editor of Current Protocols in Protein Science, as well as the Journal of Leukocyte Biology. And it's my great pleasure to welcome Dr. Abdel-Mohsen for his talk, Targetable Plasma Correlates of COVID-19 Severity, PASC Relevance. Mohamed, thank you.
Thanks, Jay, very much for the invitation and for the introduction. Let me share. I will need to, I think, an invitation to share my screen.
Yeah, let me stop my share, and that way you can put yours on.
Yes.
There we go.
Yeah, let me know when you can see my screen in full and can hear me well, hopefully.
We can see it. It's all good.
Awesome.
Like.
So yeah, thank you very much for the invitation. I'm very excited to have the chance to share some of our work with you today in COVID-19, in particular in our work identifying plasma correlates of COVID-19 severity that are targetable, and we believe that they could have a very high relevance to post-acute sequelae of COVID-19, PASC, or long COVID. And my talk today will be divided into two parts. In the first part, I will show you how we identified marker of disrupted gut permeability in the plasma of severe COVID-19 patients, and which is a work that has been published in this paper. And I will follow up on this part by a couple of preliminary data slides showing you that is how we identified several markers in the plasma of people suffering from long COVID compared to control. And in the second part of my presentation, I will show you how we identified qualitative rather than quantitative features of anti-SARS-CoV-2 specific antibodies that is also associated with both disease severity and inflammation. And again, this work has been published in this paper. And again, I will follow up on this with just a couple of quick preliminary data slides to show you how we also identified the same phenomena using antibodies isolated from people suffering or not from long COVID. So starting with the first part on disrupted gut permeability, which is the work of Leila Giron and a scientist in our lab. And let me start by telling you that is, in the last few years before the pandemic, there was an accumulation of evidence on this emerging concept of lung-gut axis. So what is that? So when there is a lung infection or damage that can cause a systemic inflammation that comes with a cytokine storm, in particular an increase in TNF-alpha and interferon gamma, those two cytokines are known to be able to injure the gut, increase intestinal permeability, increasing microbial translocation, meaning it allows microbes and their byproduct to translocate from the gut to the blood, or what is also called leaky gut. Now, when these microbes and their byproduct is being recognized by the immune system systemically, that can lead to inflammation, further cytokine storm, that can impact the original infection or damage in the lung. And eventually this vicious cycle between the lung, systemic inflammation, and microbial translocation lead to worsened disease outcome. And in some cases, when the pathogen, such as SARS-CoV-2, can also directly infect cells in the gut, that can help feed this vicious cycle. So, and that's why last year we asked the question, is there a potential link between lung, gut, and inflammation during severe COVID-19? And we started looking at this question by analyzing samples from SARS-CoV-2 negative controls and SARS-CoV-2 positive individuals that were either outpatients, which will mean that they will have a mild symptoms by definition, or inpatient, that is, we're even further divided between patient hospitalized in a normal hospital bed, so they will have moderate symptoms, or patient hospitalized in ICU, so they have severe symptoms. And we selected those samples to be all between the age of 50 to 65, so to avoid age bias in our result, and also 50% male and 50% female. And we use these plasma samples to apply a plasma system biology approach to identify gut-related targetable correlates of COVID-19 severity. In particular, we use this plasma sample from all of those individuals to measure several markers of gut-tight junction permeability and microbial translocation. We also measure several markers of systemic inflammation and immune activation. And because we also know that is not only, that is plasma metabolites and lipids not only can reflect the functionality of the gut, they can tell you if the gut is functional, normal or not, but they also in their own merits are biologically active molecules that can go and impact inflammation and immune activation. And that's why we did a full metabolomic and lipidomic analysis. We also did a glycomic analysis on those same sample, and I will tell you in details in the second part of my presentation why we thought this is very relevant, specifically to antibody-mediated innate immune function. But for the first part, I will focus on the markers of microbial translocation, inflammation, metabolomics, and lipidomics data. And before showing you some of the data, I just want to point out that is a couple of the individual in the moderate group and several of the individual in the severe group, unfortunately deceased due to the severity of their COVID-19. And I will show you their data in black dots in the subsequent slides so you can identify them. So we first looked at this marker called zonulin, and zonulin is very important because it's not just a marker for tight junction permeability, it's actually a driver for it. And as you can clearly see here, there is a very high level of this marker in the patient who were hospitalized with COVID-19. In fact, when we only looked at the hospitalized patient, we can also see that the level of zonulin is higher in the patient who eventually deceased from their COVID-19 versus whoever survived. And that was very interesting because, as I mentioned, zonulin is not just a marker of gut tight junction permeability, it's actually a driver for it. When there is an increase in zonulin that is caused by gut dysbiosis, meaning there is changes in the composition of the gut microbiome, there is an increase in zonulin, and this zonulin open the tight junction, allow microbes and their byproduct to translocate, increasing cytokine storm, increasing inflammation, increasing T cell activation, and cause loss of tolerance. And because of this, this fact, many groups even before the pandemic were interested in designing a small molecule antagonist to zonulin and use it to treat diseases that is associated with gut inflammation. And this antagonist has been used both in preclinical and clinical trials with good efficiency. So that is just an example of how our system biology approach can allow us to identify a targetable correlates of COVID-19, and zonulin being a physiological driver that has an antagonist is one example of that. But now the opening of the tight junction permeability or the opening of the tight junction should allow microbes and their byproduct to translocate to the plasma. So can we see that? Indeed, we can see evidence of bacterial translocation as measured by this marker called LBS binding protein. And you can see nicely that is people who have severe COVID-19 have more bacterial translocation than people with moderate, who have more than mild, who even have more than the control. But bacteria are not the only microbe that can translocate from the gut to the blood. The gut is actually have very high level of fungus. And the fungus, when translocated from the gut to the blood, can also induce inflammation, systemic inflammation. And we can measure fungal translocation using this marker called beta-glucan, which is a part of the cell wall of fungus. And we can measure it in the plasma. And you can see also here that is severe COVID-19 is associated with a fungal translocation, not only bacterial translocation. And both the bacterial translocation and fungal translocation should lead to myeloid inflammation, which is we can measure using markers like soluble CD14 or MPO, which are markers of monocyte and neutrophil inflammation. And indeed, as expected, you can see a very profound increase in this marker during hospitalized and severe COVID-19. Now, this is focusing in markers specifically for microbial translocation. We can also look at the different markers of inflammation and immune activation, including your usual suspect, C-reactive protein, IL-6, GDF-15, which is a marker of mitochondrial dysfunction and whatnot. And as expected, you can see an increase. Red is higher and blue is lower in this heat map. And you can see that the moderate group has more of most of these markers, and the severe even have more. But our question here was whether these markers of tight junction permeability, zonulin, LBB, bacterial translocation, LBB, or fungal translocation, beta-glucan, will correlate with this marker of inflammation. And the short answer is yes. You can see here the Spearman correlation coefficient very strong with a significant FDR. That is these markers strongly correlate with inflammation. Just to show you a couple of examples of how strong these correlations are, in the top here, you can see a correlation in the x-axis between bacterial translocation, and in the y-axis with inflammation as indicated by IL-6 level. And in the bottom is the same correlation, but in the x-axis is fungal translocation. And you can see in both cases, there is a very strong correlation between a marker of tight junction permeability, bacterial translocation or fungal translocation, and systemic inflammation. So it seems like COVID-19 severity is associated with increase in permeability of tight junction, increase in markers of bacterial and fungal translocation in a manner that is associated with inflammation. This was done in this one cohort, but we have validated that since then in multiple cohorts, including this validation cohort that was also independently collected at Rush University in collaboration with Ali Kishi and Alan Landy, of also patients with no infection, mild infection, moderate infection, and severe infection. And again, you can see the same exact data that is severe COVID-19 is associated with increase in zonulin of tight junction permeability, bacterial translocation, and monocyte inflammation. So that has been repeated in multiple cohorts. And since we actually published this data, there was an accumulation of evidence in the literature in how COVID-19 is associated with gut microbial dysbiosis, meaning changes in the composition of the gut microbiome, and microbial translocation and gut leakage that is associated with dysfunctional immune responses, higher inflammation, and higher disease severity. With the model as shown here in this review article, that is, if the gut-lung interactions I mentioned to you are resulted in inflamed leaky gut, and usually they result in inflamed leaky gut when the gut is weak, and the gut is weak usually if the person is older, obese, have diabetes or chronic infection, all confounder of severe COVID-19. If the gut is weak and this lung-gut interaction lead to inflamed leaky gut and dysbiosis, that is increasing microbial translocation and lead to uncontrolled inflammatory responses that will eventually cause a complication in various organs. While if the person has a strong immune system, a person has a strong gut, these interactions will resolve, leaving a healthy gut, preventing microbial translocation and gut leakage, maintaining controlled inflammatory responses, and that can prevent this complication in various organs. So far, I showed you data on markers of tight junction permeability and microbial translocation, but I also mentioned to you that we did a full metabolic and lipidomic analysis, because in one hand, these metabolites and lipids can reflect the function of the gut, so they can give us another layer of evidence of our hypothesis. But on the other side, these metabolite and lipids are biologically active molecule, increasing the chances that we can identify a targetable correlates. And indeed, the metabolome, the plasma metabolome during severe COVID-19 is totally dysregulated. You can see here in this PCA plot, that is the plasma metabolome of patient with moderate and severe group clustered quite distinctly from mild patients or controls. And that is due to dysregulation of several metabolic pathways, that many of them are associated to gut functionality. In fact, when we focused on the metabolite that are known to directly impacted by the gut, many of them were dysregulated, either upregulated or downregulated in the patient with moderate symptoms compared to mild, or severe symptoms compared to mild. And I want to highlight only a couple of metabolic pathway, again, because I think they are interesting and could be targetable. One of them is this one amino acid called citrulline. And I want to highlight it because the citrulline, as you can see here, has been used as a marker of intestinal function and absorption in many clinical settings. And it goes like the more citrulline, the higher intestinal function. And as you're losing it, that is a marker of loss in intestinal function. And as consistent with our own data and data of others that is actually check the microbiome in stool and gut biopsies, there is a reduction in the gut intestinal function as evidenced by less amount of citrulline as severity of COVID-19 increases. But again, and this is correlate negatively with inflammation as measured by IL-6. But again, I wanted to highlight that because citrulline, in addition to being a marker of intestinal function, it also has been shown in some preclinical, not clinical, but animal model that is to be also possibly a modulator because pre-treatment with citrulline can improve gut barriers in animal model. The other metabolic pathway I want to highlight is tryptophan catabolism. Now, tryptophan catabolism pathway and the metabolite involved in tryptophan catabolism pathway has been associated with gut microbial dysbiosis in another viral infection, HIV, when HIV is known to have also to cause also long-term complication even after viral suppression by antiretroviral therapy. Probably most of you know that is people living with HIV are still suffering from neurological impairments, cardiovascular diseases, sometimes at higher rate and younger age than HIV negative individual, even after suppressing therapy. And tryptophan catabolism has been dysregulated during HIV infection. Indeed, tryptophan catabolism is dysregulated during severe COVID-19. As you can see here, the more tryptophan catabolism, the more severe COVID-19. Individual metabolite within the tryptophan catabolism, like tryptophan itself and quinuric acid, can differentiate patient who survive versus disease. And there is a very strong correlation with inflammation. Again, I wanted to highlight this because dysregulation in tryptophan catabolism has been associated with the development and maintenance of several long-term complications during, again, another viral infection with long-term complication even after suppressive therapy, which is HIV. Dysregulation in tryptophan catabolism has been associated with subclinical atherosclerosis, liver fibrosis, diabetes, and even depression. Make you wonder if some disruption continue and maybe contribute to the long-term complication of SARS-CoV-2. But I will skip the data on lipids because of the time, but I just want to highlight that is also, as we saw with the metabolome, severe COVID-19 was also associated with quite disrupted plasma lipidome in a manner that is compatible with both gut dysfunctionality and higher inflammation. But just to conclude this part, we found that a severe COVID-19 is associated with plasma marker of disrupted intestinal integrity, higher microbial translocation, intestinal dysfunction in a manner that is linked to systemic inflammation. And these data, in addition to the data that is published by other group on using stool samples and gut biopsies showing same result, I just want to highlight that they do not imply that is a microbial translocation is the primary trigger of severe COVID-19, because likely this is a multifactorial in nature, but we think about it as these microbial translocation that can lead to inflammation, as evidenced by many studies, is kind of like a gasoline added to a already existing fire. Makes it very hard for the immune system to control this inflammation and can tip the person into a severe COVID-19 or even death. And there are many questions we are trying to investigate now. And one of the questions we're trying to investigate, obviously relevant to this group, is does the disruption in intestinal barrier integrity persist, at least in part, and maybe contribute to long COVID or PASC? And I will show you a couple of very preliminary data, just like very at the food for thought more, because we haven't done the formal analysis, but we are doing some of this work in collaboration with the long COVID clinic at UCSF and Rush University. And the data I will show you is mainly coming from sample from the UCSF from the long-term impact of infection with novel coronavirus, LINC cohort in collaboration with Steve Deeks, Michael Beloso, and Tim Henrich, where they send us samples from well-characterized 56 individuals who are 4 months post-convalescence, not suffering from any symptoms, or 61 individuals who are suffering from long COVID, suffering from PASC. And we ask as a question, does some of those markers we observed during severe COVID persist or not? And here I'm showing you some data on fungal translocation. And as you can see clearly here in the left side, that is the patient with PASC have higher fungal translocation compared to control. The patient with PASC are in blue and the control in gray. And even when we take actually those patients with PASC and divide them into patients who have 1 to 7 symptoms or more than 8 symptoms, you can see that this PASC is associated with increase in a fungal translocation, and it seems that is associated with the number of symptoms that this patient are suffering from. We didn't see as much striking increase with bacterial translocation. There is a trend of an increase of bacterial translocation, but it's not as clear, as dramatic as the fungal translocation. So it seems like PASC is associated with more of a fungal translocation than bacterial translocation for some reason. We haven't seen any difference in level of soluble CD14. Again, it's a marker of myeloid inflammation. And we would expect that is both bacterial translocation and fungal translocation correlate with more tight junction permeability. Indeed, in these individuals, tight junction permeability in the x-axis correlate with fungal translocation, and the fungal translocation correlate with bacterial translocation. So again, seems like, again, preliminary, that this PASC is associated with an increase in fungal translocation, and there could be 2 explanations of these data. In one explanation is that, again, fungal translocation has been associated with immune activation and inflammation during, again, another viral infection that is associated with long-term complication, HIV. But fungal translocation is not just a marker of inflammation, is a driver for it, because beta-glucan, which is a cell wall of the fungus, can bind to its receptor called dictin in the surface of myeloid cells, and that's enough to induce inflammation by activating inflammasome. So one can imagine or envision that is possible that is even following recovery from acute COVID-19, some level of fungal translocation persists in some individual, and does this fungal translocation contribute to PASC by inducing chronic inflammation? But there is another explanation of this data. And the other possibility is that this could be just a reflection of the health, metabolic, or socioeconomic status of the people prone to PASC. And the reason why I'm highlighting this other possibility is I'm showing you here data from another cohort in quote healthy individuals, even that is with pre-pandemic. And you can see here in the left that is a level of fungal translocation in the y-axis correlate with age. It's not a strong correlation, but it does correlate with age. As you're getting older, you have more fungal translocation. But even after you adjust for age, gender, and ethnicity, you can see people who have, again, quote healthy, but who have metabolic syndrome, have more fungal translocation compared to the people who are not having metabolic syndrome. And again, people who also have a lower socioeconomic status, and there is obviously many ways to measure that. I'm just showing you one way people referring to it, which is education. As people having lower education, they do have more fungal translocation. So for some reason, it's unclear why for us. But it's possible that it's people who are older, with pre-existing metabolic syndrome and all lower socioeconomic status are more prone to both fungal translocation and PASC, regardless if these two things are linked or not. And obviously, as I mentioned, these are very preliminary data. Take it with a grain of salt. I wanted to show it as a food of thought, but we are currently doing our multi-omic analysis on the same sample from UCSF cohort, and we are obviously doing a formal analysis correcting for all of those different confounder using a multivariable analysis, and we will know the direct and indirect impact of these confounder that is known to be associated with both disease severity and long COVID progression on the beta-glucan. But also there is other questions we are investigating, like obviously we focus in targetable correlates for a reason, and we ask the question, can modulator of tight junction permeability lessen either disease severity or PASC? And that is something we are doing with Jay at Tulane National Primate Research Center. A very important question, I mentioned to you that the SARS-CoV-2 can infect the gut, but also the cytokine that is produced by SARS-CoV-2 infection in the lung can also injure the gut. It's not clear if SARS-CoV-2 infection directly through direct infection or indirectly through its cytokine storm disrupt the intestinal barrier integrity, and what will be the impact of an established gut disruptor like alcohol abuse on post-acute sequelae of COVID-19 is something we are doing now, or trying to do using gut organoids in collaboration with Ali Kishad, the Russian university, who is an expert in gut organoids, as well as on disruptor of gut permeability, in particular alcohol abuse. And last but not least, I showed you some dramatic effect in severe COVID-19 on some of our markers. And one can also ask as a question, can some of these markers we identified predict the risk of disease severity or PASC if measured before or immediately after diagnosis? And we are exploring that in the general population, but also in a special population of HIV-infected individuals, because those individuals already have gut leakage to start with. And one can imagine that is maybe they will be more prone to long COVID. And we're doing this using samples from the Max and Wise cohort, which is a big national cohort collecting samples before and after pandemic, periodically in collaboration with Todd Brown at Johns Hopkins University. And we're hoping that is the answer of some of these questions will allow us to better understand the pathogenesis of PASC and hopefully even identify both biomarkers and targets to treat it. And that will conclude the first part of my presentation, and I will be ready to go to the second part. But I'm wondering, Jay, do we stop and take a question for the first part now, or should I continue and take all the questions at the end?
Yeah, let's take a few questions. You know, I think that first part was so interesting, and I'm sure it's going to stimulate many questions. So let's just open this up for a few questions on this. I don't know, is there, if anybody wants to ask a question, or I don't know if people are unmuted. I can ask a question myself. Josh has yeah, put the question answer, the Q&A in the on the lower bar, and you can put your questions there. While you're doing that, you know, there's so many questions here, but I was very interested in you know, what you were saying about tryptophan catabolism, you know, and it's also, seems to be involved in other infections, also aging. Have you looked at the other end of this? I mean, I know tryptophan catabolism is the, let's say, de novo pathway to make NAD. Have you looked at the salvage pathways or price handler pathways to make NAD and what's going on with that? Or have you looked at catabolism of NAD itself?
We haven't actually, but that will be interesting. We did a full metabolic analysis, so the data is there. So we can definitely look into that, into NAD metabolism, because obviously, again, that is important for many diseases, including long-term complication of HIV. Yeah, we'll look into that.
Great. So there's a couple of questions on the chat. Prabha Fernandez is asking, have any antifungals been tested in post-COVID?
Not that I know of. I think that will be interesting again, but I don't, obviously there is many different things tested for COVID in the last 2 years. It's very hard to keep track with everything going on, but not to my knowledge, at least.
Okay. So I guess Ashwin Bolunkapal say should neomycin be used, oral non-absorbable antibiotic, or is that a suggestion? Yeah,
I think that is a very important question. Now, the antibiotic is actually increases microbial translocation. Sometimes depends on the antibiotic, and a lot of the patient, the data I showed you in this cohort were the sample were collected immediately after the patient were administered to the hospital or the ICU before they being even treated. But a lot of those people will go and be treated with antibiotic, with heavy antibiotic. That is part of the course of at least when those samples were collected. And those antibiotic might actually worsen gut permeability. But this didn't impact this result I show it to you. But that is one thing that is we would say about this data, that those antibiotic treatment should be done carefully, because if this microbial translocation and disrupted gut barrier is contributing to disease severity, this antibiotic course might actually makes it worse. Again, depends on the antibiotic and how it's being administered. But as I said, all of those samples were collected immediately after the patient was admitted, before they were treated, because not only antibiotic, but also steroid and other things that might impact the results. So this was before that.
Okay, this has been great. I mean, already you see some new potential therapeutics. There's one comment: there are studies that show an association with bacterial translocation changes in the gut microbiome. Do you know if anyone is looking at the impact of using fecal transplant and other means of changing the gut flora on PASC?
Yeah, again, I don't know of any trial going like this. I'm sure all of you know that is while fecal transplantation worked very well with the C. diff infection, it didn't really, hit the nail with something like HIV, for an example, so far. So I'm not aware of any studies did that, but maybe something, as I said, going to the root, going to something like zonulin, which is actually the physiological relevant driver, might be a little bit better than trying to do a fecal transplantation, because there is a lot of moving parts with fecal transplantation. But again, I'm not aware of anyone did such a study, but it will be interesting to see, because fecal transplantation working in some context, again, as C. diff very well, but in some other context, like HIV, for some reason it doesn't work. And I don't think it's clear why.
Okay, and someone else says anonymously, will the MAX and WISE cohort be able to compare PASC and markers of PASC in persons with and without HIV?
Yeah, so that is something we just starting with with support of the Campbell Foundation. But the Max and WISE collecting samples anyway from HIV negative and HIV positive over time, so they have sample immediately pre-pandemic. So we are now in the process of receiving the samples, and yes, the idea is to compare the HIV negative to the HIV positive, specifically with the fact that is HIV infected individual or like some of them at least, have a pre-existing condition of leaky gut. So will that increase their chances of having PASC or not? And if that is related to this marker of type of the microbial translocation, and will we see that also in the HIV negative? I think that will be very interesting thing to see in the next few months. But yeah, we will compare both with and without HIV for sure.
Great, great. So let's go on to the next part.
Absolutely. All right. So the second part, I want to actually go back, is focusing in antibodies, SARS-CoV-2 antibody, and how we identify it qualitative rather than quantitative difference. And this work is work of Samson Adeniji, a postdoc in our lab. And our lab actually at Worcester, as Jay mentioned, doing a lot of work in trying to understand the role of host glycans in innate immune functions during long-term viral infection. And the reason why we do so is because it has been clear recently, not in infectious diseases field, but other field, that is the glycans in circulating glycoproteins, including antibodies, can initiate a strong anti or pro-inflammatory response. And that is depends on the glycan structure and by binding to sugar binding proteins called lectins, expressed in pretty much almost all immune cells. And it has been shown that these phenomena contribute to the development and maintenance of several inflammation-associated diseases or comorbidities in the general public. And that's why we are very interested in understanding how this phenomenon contribute to the long-term, to the development of the long-term complication during viral infection. And we have been mainly working in HIV infection. And if you are interested in this line of research, you can read more about it in the different publication we published in the last couple of years on it. And I will give you, before I show you some data on SARS-CoV-2, I want to give you one single example on HIV. And indeed, again, HIV is another viral infection that is, despite viral suppression by antiretroviral therapy, is still associated with long-term complication. And one of those complications is neurological impairments. And indeed, we found that there is a disruption in the gut, in the plasma glycome, in HIV infection in infected individuals, and this disruption does not resolve to full normal even after long-term suppression with antiretroviral therapy. In particular, there is a reduction in anti-inflammatory glycan, and again, in a manner that is not fully reversible by antiretroviral therapy. And we thought that this disruption in the plasma glycans can differentiate patient with and without neurological impairments, and its degree correlate with the degree of neurological impairments in these individuals. But very importantly, this actually can be fixed using, in collaboration with David Volosky in Mount Sinai and using his mouse model of HIV infection on neurological impairments. You can see that is a mouse infected with HIV here in yellow are making more errors and taking longer time to reach to their object in this water maze experiments compared to the HIV negative mice here in gray. But if you treat this mice with a special inhibitor for this glycomic degradation that is we made here in red, you can see that we could revert neurological impairments to absolutely normal level. So that's just an example of how circulating glycans can be a marker and a driver of inflammation during long-term complication of viral infection. But in addition to their impact on inflammation, glycans, specifically those on antibody and IgG, is playing a very important role in regulating innate immune function. I'm sure all of you know that is beyond neutralization. Antibodies can engage many innate immune cells to elicit important innate immune functions in our way to fight viral infection. And this is include antibody-mediated cytotoxicity by NK cells, antibody-mediated phagocytosis by myeloid cells, and the deposition of complement in order to elicit complement activation. And as also you know, while these markers or these functions are important to fight viral infection, they can also in their own merit causing inflammation, like complement activation is known to be associated with inflammation during SARS-CoV-2. Now, glycans are playing a very important role in the ability of the antibody to elicit this function. For an example, the sugar here called fructose, dramatically inhibit the ability of antibody to elicit cytotoxicity by NK, while the sugar called galactose is important to induce phagocytosis and complement deposition. And again, we have been studying this during HIV, and just showing you one quick example that is even after long-term suppressive therapy, people with HIV still some of them suffering from subclinical atherosclerosis. And when we did a glycemic analysis on the IgG from individual with and without subclinical atherosclerosis, you can see there is a reduction in the level of this sugar called galactose, which is, I mentioned to you, not important for NK functionality, but very important for phagocytosis and complement deposition. And indeed, when we actually take these antibody and functionally test their ability to elicit HIV-specific cytotoxicity, phagocytosis, and complement deposition, you can see no effect on NK functionality, but you can see a change in their ability to elicit phagocytosis and complement deposition. Again, just an example of how these glycans on antibody are important for this innate immune function. Now, I told you we did, that is why we did a full glycemic analysis on this cohort of COVID-19. And indeed, we find that a severe COVID-19 is associated with disruption in the plasma glycome, changes in the IgG, in IgA, in total plasma glycome. And that is actually promoted us to measure the same innate immune functions in the plasma of those individuals. And here I'm showing you the ability of S1 specific antibodies and RBD specific antibody to elicit complement deposition. You can see that clearly that is antibodies from hospitalized patient were able to elicit complement deposition much more than non-hospitalized patient, both S1 specific and RBD specific. And interestingly, that is with the opposite with phagocytosis. The S1 specific antibodies from patient with hospitalized with COVID-19 were actually less able to elicit phagocytosis. So it seems like that is the COVID-19 severity is associated with differential antibody Fc-mediated immune function, in particular higher level of complement deposition from hospitalized patient, which is consistent with the idea that those patients have complement activation, which has been already shown to be a potential driver for severity during COVID-19. And very importantly, this was not dependent or at least fully explained by the titer. When we measure spike titer or RBD titer in this individual, you can see there is absolutely no difference in the titer of a spike or RBD between hospitalized and non-hospitalized patient. It was also not fully explained by the neutralization. Again, using pseudovirus neutralization assay, you can see that is the antibodies from hospitalized and non-hospitalized were equally able to neutralize the virus. So it seems like there is a qualitative difference beyond quantitative in this antibody, which is might be, but we don't have a proof of that, might be related to their glycosylation, that is makes them less or more able to elicit these innate immune function. That is while might be important for viral infection, they can also cause inflammation, specifically complement deposition. And indeed here in this correlation heat map, when we correlate here the different function, and in the column, we correlate different markers of inflammation and immune activation. And in this correlation heat map, red is a positive correlation and blue is a negative correlation. The ability of S1 specific antibody or RBD specific antibody to elicit complement deposition indeed correlates strongly positive with inflammation in this individual. So this short part conclusion is that is, we found differential qualitative rather than quantitative features of anti-SARS-CoV-2 specific antibodies that is elicit specific innate immune function that is associated with COVID-19 severity. And in particular, we found that is the antibodies from hospitalized patient were able to elicit more complement deposition despite similar titer and similar neutralization ability compared to antibodies from individual with mild COVID-19, and this is linked to inflammation. And we think that is understanding these qualitative features of anti-SARS-CoV-2 specific antibodies will be very important beyond neutralization, beyond titer, will be important to develop monoclonal antibody therapeutic and vaccines with optimal balance between efficacy and safety. You might want higher neutralization, higher titer, maybe higher phagocytosis, but less complement deposition if you want to avoid any inflammation. Again, it's not clear, but I think it's very important to understand and study moving forward. And again, relevant to PASC, and again, the samples from the ANC cohort from UCSF, we also ask us the question, how these antibody qualitative feature correlate with the with COVID-19, long COVID. And again, here I'm showing you the titer of a spike in the individual with long COVID, again here in dark blue versus the people without long COVID, even after they were infected with COVID-19. But after convalescence, 4 months of convalescence. And there is some, it's not like not below 0.05, but it's clear trend of lower actually titer of a spike protein in individual with long COVID compared to control. This was also true in RBD titer. Again, make us believe this is probably true, even though it didn't reach statistical significance. There is a lower titer of anti-spike antibody and anti-RBD antibody in these individuals. But despite the lower titer, for some interesting reason, when we normalized to this titer, these antibodies were able to elicit more S1 specific phagocytosis, spike specific complement deposition, and spike S1 specific complement deposition. And you can see that in each one of those cases. Same thing exactly we see with RBD. Again, despite lower titer, the persistent antibody is somehow eliciting more innate immune function. Again, it's not clear for us why is that. And again, it's totally preliminary data, but it seems like individual developed PASC might have lower titer of spike and RBD antibodies, but the antibody that do persist are more likely to initiate complement deposition and phagocytosis, maybe inflammation via FC receptor. And this is the language of Steve Deeks when he saw this data. And but as I mentioned, I think this understanding this qualitative rather than quantitative features of the antibodies, both during severe COVID-19 and PASC, both naturally induced antibody and vaccine mediated, I think will be very important in order to make an optimal balance between efficacy and safety. And with that, I just go to my acknowledgement slides. And obviously, I showed you data from Leila and Samson, but I would like to thank everyone else in our lab, our collaborator, specifically our collaborator at Rush University, Alan Landy and Ali Kish, and UCSF, Steve Deeks, Tim Henrich and Michael Beluso. And most of the work I showed you was supported by this supplement from NIDDK, but also some of the ongoing work is being funded by the Campbell Foundation, specifically the work on HIV. And thank you for your attention, and we'll be happy to take any further questions.
Thank you very much, Mohamed. That was really, really exciting. You know, it just really, really amazing work. While we're waiting for some other questions to come up in the chat, you know, have you looked at any, you know, for what the drivers are of changes in complement deposition or increased complement deposition versus phagocytosis? Are there any, let's say, correlations between ADCD and, let's say, zonulin, beta-glucans, or tryptophan catabolism as drivers?
There is, but I don't know if actually Steve Deeks is in the call, but he always hates when we try to connect these two stories together. Like I do actually believe that is the microbiome is releasing high level of glycan degrading enzyme that can translocate and degrade glycans in IgG. And we did see that is the IgG is disrupted, the glycan disrupted quite significantly. And actually, this has been also shown by other cohort in Europe. There was a several science paper and science immunology paper showing that the IgG glycosylation is quite changes during COVID-19 and associated with severity. I think that is established fact. What is the driver of it? Whether this is related to indigenous enzyme that is produced by the body, or maybe exogenous enzyme that is produced by the microbiome and by the open tight junction translocate to the blood and causing the changes. That is like a far-fetched hypothesis, and that's why we try to separate these two stories for now. But it's totally possible that they are linked. We do think that is this difference in the qualitative feature of the antibody is related to the changes in the glycosylation, but also that opens the question, what is causing these changes in the glycosylation? And that is like an ongoing work in our lab, because we, as you saw, we're doing that for both SARS-CoV-2, but also HIV, and we see similar result.
Okay, we have a couple comments, questions from Prabha Fernandez. Complement inhibitors have been tested in COVID, in severe COVID. Have these patients, survivors, been followed to see if they develop PASC?
If the samples from the UCSF cohort of Steve Deeks, they already have data on the hospitalization and also HIV infection, and that will be part of our multivariate analysis when we analyze all of this data together when we're done with the multi-omic approach. The same thing with the Max and Y's. The thing with the Max and Y's is they do have samples even because they already collecting samples. It was before every 6 months for the last maybe 30 years. And I think now it's moved to every year. So they have samples before the pandemic. And they, through surveys, they know the disease severity and the likelihood to develop PASC. So I think in the next few months, will be more clear, but I think in both of these two cohorts, there will be a good longitudinal data in both severity and PASC that will help us dramatically to understand this, the longitudinal kinetics of these correlations.
Great, and Mark Esser has a comment and question. Very nice presentations. Have you or others been able to mimic more or less severe disease and or long COVID in animal models?
I think that's a question for you, Jay. Right?
Yeah, we're working together on that. Hopefully we will.
I refer to you on that. I think there are some aspects of the disease has been mimicked in different animal model. And I think we, like Jay and I and others, are working to try to see if this particular aspect can be mimicked in which model, so we can actually try to, interfere with it.
Yeah, I mean, particularly we know, let's say tryptophan catabolism, we know in non-human primates and also mice. I mean, there's probably other things we need to look at in terms of the ADCD and zonulin and some of these other things, and maybe even the potential of antifungals to be effective. But these are great future directions. Yeah. So I don't see any more questions. That was really wonderful presentation, very provocative. So many lines of investigation can be opened up, I think, from consideration of your work, and just really enjoyed this. So thank you so much.
Thank you very much for the invitation for you and Josh, and I really enjoyed it. And yeah, if anyone has any ideas for collaboration, we are always happy to collaborate. Thank you very much.
Thanks. I'm going to share my screen again just to go on to, just what's coming up next. Let's see if I can get this. Whoops. And this, just to show what our next two, what our next two talks are. The next one is Dr. Akiko Iwasaki, that Dr. Fessel will be introducing that one, on 10/28, and on 1/4, Dr. Shelley Farhadian on neurological symptoms during and after COVID-19 and some CSF markers. So, and we're probably going to be adding some more talks to this symposium. It seems to be extremely valuable and I think insightful, and I think that this is a very interested group, and it seems to be growing also as we go, and I think it's just generating a lot of excitement, from the NIH community, the academic community, pharma. I just think it's really fantastic, and I want to thank everyone who's attending. Thank the speakers. Thank you, Mohamed. This was just a great talk, and I look forward to further, discussions, further seminars, and also just so everybody knows, to reiterate, these talks will be available in recording for whoever's registered to see them, and I know I'll be looking at these multiple times. So thank you again, everybody, so much.
And I want to echo that. Thank you, Jay, and thank you, Mohamed, and everyone for attending. Yeah, this is the highlight of my week every time we have one of these.
So true. Okay. Well, we'll sign off. Thanks again, everybody. Take care.